Today, our guest speaker is Dr. Oscar Ferrante from the University of Birmingham. And Oscar is one of those rare speakers who can really give us some insights into research results that we are also extremely interested in. So it's about consciousness, and a lot of us here work with on consciousness or related topics. But at the same time, the methods that Oscar uses are super interesting, not just in scientific terms. So, for example, he's also a contributor to standardizing MEG analysis pipelines, but also in kind of meta-scientific terms. So in terms of open science, adversarial collaborations, data management, and so on. And I hope we get some important insights into all of these aspects today. So Oscar finished his PhD a few years ago in Verona, in Italy, in the group of Leonardo Chelazzi, where he mostly worked using psychophysics and transcranial magnetic stimulation on attention and its relationship with statistical learning. And in 2019, he joined the neuronal oscillations group led by Professor Ola Jensen in Birmingham, where he now mostly works with MEG on sort of two main topics. One is consciousness in this big consortium called the Cogitate project, and also he continues now with his work on statistical learning and attention. So yeah, without further ado, Oscar, the floor is yours, and I will handle questions as they arise. Thank you.
Thank you so much for the nice introduction, Lisa. Yeah, so today I'm going to talk about this adversarial collaboration called Cogitate. That is, we run to evaluate different theories of consciousness. I will start with a very brief introduction about what is consciousness. This is a question that has been for millennia on the mind of philosophers and thinkers. So a definitive definition is also difficult to provide because of the different schools. But one definition that I like, that it's more cognitive, also based on my background, is the awareness, so like what it feels like, of an internal or external stimulus. So apart from the philosophical and discussion of consciousness, this is also a topic that has been discussed a lot in psychology, especially during the period of Sigmund Freud. And then there was a period in which it was difficult to discuss scientifically about consciousness until around 30 years ago. There was a seminal paper published by Crick and Koch, in which they opened the exploration of consciousness from a neuroscientific point of view. So the question that they raise is, what are the neuronal correlates of consciousness? And this is what we are interested in Cogitate. So there are many theories that have been proposed to explain the neural correlates of consciousness or consciousness in general. In our collaboration, we focus on two theories. One is the global neuronal workspace theory. This theory proposed that a stimulus is processed in a subliminal way until a certain point, but only when this information reaches the so-called global workspace, then we become conscious of this stimulus. And what happens is that the global neural workspace, which is localized in the frontoparietal area, broadcasts the information to the rest of the brain. And this broadcasting is what is considered the conscious event. And this theory was proposed by Barr and then moved to the neuronal space by Stanislav Dehaene, who's part of the Cogitate project. Not the theory that we investigate in our project is the integrated information theory. This theory starts from a phenomenological point of view, describing the experience itself and its basic properties, and then trying to apply this to the brain, looking at the same properties in the brain structure. And Giulio Tononi and his colleagues found that the neurons in the so-called major complex, it is mainly localized in the posterior part of the brain, has the properties to contain this integrated information, which is for IIT the phenomenal experience of consciousness. But these are only two of many theories of consciousness, and here there is a very good review paper by Seth and Bain, published a couple of years ago, discussing all the major theories. But the problem is that there are too many theories, and then what happens in this case is that theories start working in isolation in the so-called echo chambers, And as shown by a meta-analysis published a couple of years ago by Joran and colleagues, what happens is that at the end, theories will work on their own theorists will work on their own theory, and then focalize on providing support to their hypothesis, then reducing the chances to find novel discoveries. So in order to fix this problem, what we propose is to try to put these theories together in the so-called adversarial collaboration. So putting them together in a situation in which they have to talk with each other, try to find a way to test the two theories or more theories together using the same methods, same experiments, same data. And this is what we do in Cogitate. And as you can imagine, to do something like big like this, we need a lot of people. And here you can see all the members of Cogitate, and especially the organizer of the project, Lucia Meloni, Liad Mudrik, and Michael Pitts, together with Stanislav Leal, Giulio Tononi, and Christoph Koch. So in Cogitate, we put very emphasis to open science. So for us, it's very important to create a team-based science. That's why we have so many people, and also from many different countries and universities. And also we use this approach called adversarial collaboration. As I said, is the main way to test different theories, in our opinion. We also use different brain imaging methods, specifically MEG, fMRI, and intracranial EEG, in order to get data from with different temporal and spatial resolution and provide a better answer to our experimental question. It's also important to have a good sample, and that's why we have a high-powered study with more than 250 subjects across 6 different labs, 2 for each of the 3 modalities, in 5 different countries. And we also, in order to collect data in 6 different labs, we have to work on standardized material. And one thing that we also are very focused on was the replicability of our findings. And what we did in this, from this point of view, is to divide the analysis in 2 phases. The first phase called optimization phase, in which we only look at one part of the data and developed our analysis methods. Then, once we were happy with what we had, we pre-registered the methods, explaining all the details of the analysis that we were gonna do to test the predictions, and only then test the prediction on a separate and bigger sample in the replication phase. So this project has had many challenges. Now it's been more than 5 years that we've been working on the project, and we realized that many of the issues were related to the beginning of the project, or starting to get to the results. Here I want to talk a little bit about 3 challenges. One, which was related to ensuring that the data had a good quality, and I will talk about the standardization and data quality checks. The second challenge was about the validity of the experiments themselves, so whether they were testing what we were interested in and providing the information that we needed and the conditions to test the hypothesis. And then the third challenge was about translating the theories into empirical tests. And I will talk about the case of connectivity to explain the difficulties that we encountered in this part of the project. So regarding the first challenge, so ensuring the data quality, so we had to work in order to make our experimental protocols standardized across different labs and different modalities, and make sure that the quality of the data were good from different points of view. And also in terms of data analysis, we have to make sure that our analysis could be reproduced by also people who were interested in trying different approaches. So in terms of standardization, all the work that we did in the preliminary phase of the project is now part of a paper that is going to propose different way to test the experiment before collecting the data. For instance, by looking at the log file, so whether all the information that we need are available, or triggers in case of MEG, intracranial EEG, and fMRI, so whether the neuroimaging data contains the information that we need in order to conduct the analysis. For instance, the information about when a stimulus is presented, and also other tests related to the duration of the stimulus presentation, and if the stimulus was presented exactly at the time in which we wanted it to be presented. And also other parameters like the size of the stimuli were standardized across different labs. So we also have to develop methods in order to make that possible. Regarding the quality of the data, we developed 3-level checks, quality check. The first level, in which we start from basic tests, looking, for instance, whether the data are correctly anonymized, because from the open science point of view, we also want to release all the data and all the analysis codes that we're producing. We want to make sure that when we release the data, they don't contain any personal information. And also whether the data are complete, of course, contains all the information that needs to be, and also consistent. So because we also want to share the data in a way that they can be analyzed, and we follow standards like the BIT standard to make sure that the information can be understood from an external user. Then we have a second level of quality checks related to the additional data. Here in this case, it's referring to the behavioral data. And here we look at whether the participant was performing the task in the correct way, and whether the participant was paying attention to what was presented in front of them. And then there is a third level, the final level of quality checks, in which we were looking at the neural data. And for instance, checking whether the level of noise was good enough to use that, to include that specific data set in the analysis, or also other basic modality specific analysis, like for instance, in MEG, looking at evoked responses after the stimulus presentation, signals that the data are of good quality to test the predictions. And then we also focus on what the way we analyze the data. So we discuss a lot about how to pre-process the data and then move to the analysis itself. And this discussion produced a paper called FLAX, which is a pipeline for MEG data analysis, in which we describe all the steps and we share codes and tutorials on how to analyze MEG data from raw data to source modeling, time frequency representation, and decoding analysis. And this is a project that started with Cogitate, and thanks to the fact that we had different labs and we have to at the beginning we were conducting analysis in a different way, but then we have to work together, and we had to find a way to analyze the data that was good for all of us. And then thanks to this discussion, we released the pipeline. And also this year, we're starting the first summer school that we call the FLAX Toolkit, that will be here in Birmingham in June. Unfortunately, now the call is closed, but I hope that we will be able to organize another one next year and in the following years. So in case you're interested in learning MEG data analysis, please keep an eye on this. Okay, so now we check that the data are good, that we're analyzing the data in a way that makes sense for us. Then let's check that the experiment itself does what it's supposed to do. So in Cogitate, we have 2 experiments. In this presentation, I will focus only on the first experiment, and I will briefly mention the second experiment at the end of the presentation. So experiment 1 is a visual detection task. So for the beginning of each trial, 2 target stimuli are presented in different orientation. Like here, you can see this is a trial object, a phase object trial, and you can see here that there is 1 phase into different orientation and an object clock here into different orientations. And these are the 2 targets for that trial. Then we present a series of images to the participant, and the participant has to press a key when one of the 2 target images is presented. Like in this case, this is the target phase, and the participant has to press key. Sometimes we present stimuli of the relevant category, like in this case a phase, but that is not a target. And then in that case, we will consider this a non-target task relevant stimulus. Then we also have other categories like this letter that is not category that is relevant for the task. Then we will define this as task relevant. So in total, we have 4 categories: phases, objects, letters, and fold fonts. And on different trials, we have either phase and objects as relevant or letter and false ones as relevant. But the task is always the same, to detect the target stimulus. Importantly, we also manipulate the duration of the presentation of each of the stimuli. So each image is presented for either half a second, 1 second, or 1 second and a half. And this is important for one of the analyses that I will explain later on. But this experiment was designed with 3 core features. The first is that all the stimuli are presented in a supra-threshold way, meaning that they are all undoubtedly conscious, so the participant should perceive all of them in conscious way. Second, that by having task relevant and task irrelevant stimuli, we can also look at the difference between these 2 conditions and look at any effect related to task relevance, which is important in order to avoid confounding effects of related to consciousness with other executive functions. Then the third core feature is that, as I said before, there is a presentation of the stimuli manipulated in terms of duration. So we have 3 different durations, and this is also important for testing the prediction of the theories. But then we ask, does the task serve its purpose? Then we have to look at the results of the behavioral performance. And we found that in all modalities, MEG, fMRI, intracranial EEG, there was a good level of accuracy with very low false response, and that the difference between the modalities and the labs were very marginal, with worse performance in the case of intracranial EEG. But this makes sense because here we have clinical patients who have to do this task during what they are implanted with electrodes and then in a setup, in a clinical setup. Okay, then from behavioral point of view, we can say that the subjects were conscious of the stimuli, and also then when we look at the eye tracking data, we found that there were similar fixation patterns for the 3 different durations, and also that this didn't differ across the 3 modalities. Then any difference in terms of brain activity in this case cannot be explained by the different fixation patterns. But then we also asked whether the participants were perceiving all of the stimuli. And here I'm showing an example of one trial in which phase and objects are the task relevant categories. And then the target phase is presented. So that if the participant can respond to the target, then we are quite sure that the participant was aware of this stimulus. Then we have a non-target stimulus, but of the same category, which means that in order to not respond to this stimulus, the participant have to process the phase and recognize whether it's the target phase or not. Then also here we can assume that the participant is aware of the specific phase. While for the task irrelevant condition, the participant in this case sees a letter, but do we know whether the participant is aware that this letter is a K, or is the participant just rejecting anything that is not a phase and an object? So in order to answer to these questions, Alex did a control experiment, behavioral experiment, in which he added 1 second part to the experiment, in which after a delay, he presented the participant with a surprise memory task, asking them, presenting to them the different images and asking, did you see this image before? And the results here shows that for the relevant and irrelevant, there wasn't any significant difference in terms of performance. So this answering the question that the participants were aware of the stimuli and that there was no difference between a relevant and irrelevant stimulus in terms of awareness. Okay, then now we validated also our experiment. The next step is to go to the theories and translate their predictions into something that we can test in the lab. And I will talk about a specific case, which is the one, the connectivity one. What I'm showing here is part of the very long pre-registration that we wrote. And this is what the 2 proponents, Stan and Giulio, wrote about what they expect in terms of connectivity pattern. So we can have a look together. And so they say that the theories make different predictions regarding the connectivity patterns and interactions among cortical regions necessary for conscious perception. GNW, so let's mark this one, postulates that global information sharing is mediated by a metastable state of long-range interarea interactions, including prefrontal areas. Okay, so we need prefrontal areas and category-specific areas. Okay, so we have some ingredients here. So let's continue and go here. IIT, on the other hand, claims that the substrate of consciousness is a maximum of integrated information presumed to be primarily located in the posterior cortex. So we need to look at the posterior cortex. Then I will skip here. Each distinct conscious experience should lead to distinct patterns of synchronization between areas in posterior cortex, for instance, category selective areas and early sensory areas. Okay, now we have also the ingredients for IIT. Okay, let's try to create a scheme explaining what we need to look for. Then we have for GNW prefrontal cortex, for IIT early visual areas, and for both we have category selective areas. So because they predict that the synchronization is higher when a category specific stimulus is presented, then let's have two category selective areas and manipulate the stimulus presentation. Let's say that we have a phase, and then we look at the phase area. So for GNW in this case, we should find strong connectivity between prefrontal cortex and the phase area. While for IIT, there should be strong connectivity between early visual cortex and the phase area. Now let's add a second area, a second category, let's say objects. Now we have another prediction here. For GNW, strong connectivity between the object area and prefrontal cortex when the object is presented, and the same for IIT, but this time between early visual areas and object areas. Now we have our nodes and connections, and we can start thinking how to test it. So here the challenges are also three. The first one is to define what GNW and IIT mean with prefrontal cortex and early visual area. The second is to identify these content-specific areas. And the third challenge is to quantify the brain synchronization. So how to find these connections in the brain. I'll start with the first okay. With the first challenge, how to define GNW and IIT core neural correlates in the brain. And this work was done by Jamil and Alex. And what they did is to look at the literature and discuss with the proponents, and then look at the brain and using an atlas to define areas that were included in the GNW prefrontal cortex and areas that were included in the IIT posterior cortex. And then we end up with this nice representation of, in green, we have areas in which we should find a pattern predicted by GNW, in blue, areas in which we should find a pattern predicted by IIT. And then in the case of connectivity, we only look for IIT at the V1, V2 early visual cortex. Okay, now we have defined this part. Then the next is define the content-specific brain areas. So what we did in the EEG, so what Alex did in the EEG is to look at the difference in activity between phase and the other stimuli, and also between objects and other stimuli. And then looking at each electrode inside the brain and finding electrodes that were selected for phases, meaning electrodes showing activity and increasing activity only when a phase is presented, like in the case here, and also defining other electrodes that were responsive to objects, like this one here. And then this way to have also the category-specific brain regions. That approach is not possible with MEG because of the spatial resolution. So we have a more spread activation. We don't have the same definition as intracranial EEG. And then we use a different approach, which is based on the same contrast, creating spatial maps, spatial filters that can be applied to the electrodes, to the sensors, to the MEG sensors, in order to maximize the activity for a specific category. Like you see in this case, we have a spatial map for phase selective activity, and we see a stronger evoked response in blue for phases than for objects. And then we did the same for objects, creating the object spatial filter and finding now the spatial, the evoked activity stronger for objects than for phases. Then now we have to move to fMRI. Unfortunately, this approach using this contrast didn't work for fMRI, so we have to do something different. And what Jamil did is to directly compare faces and objects and looking at the distribution of the voxels in terms of the difference between the activation for faces and objects. And then using the voxels in the two tails as object-specific and face-specific areas. And then when this is reported on the brain, we see here that when focusing on the fusiform area, there are some voxels here marked in red that are selective for faces. And in the image below, we have in red the voxels selective for objects. So now we also have the content-specific areas for all modalities. What is left is how to measure connectivity in the brain. So in this case, there are many methods. And for intracranial EEG and MEG, we decided to use two methods, one based on coherence, which gives an idea of the synchronization in terms of phase. So whether the activity, rhythmic activity of different areas are in phase or not in phase, and another method based on mutual information, which gives us an index of power-based synchronization, which means that when two areas are more active at the same time, those two areas must be communicating with one another. Because of the low temporal resolution of fMRI, we cannot have a phase-dependent measure, and then we decided to use the generalized psychophysiological interaction, which works by defining each trial based on the condition, based on what stimulus was presented, and then trying to fit the bold activity to this to the different conditions, and finding voxels that show the same modulation, meaning voxels that are active or suppressed when the specific condition is presented. Okay, now we have all the ingredients. It means that we can proceed to test the theories. So we have good data, the experiments are working, and we now know how to test the theories. So let's move on. It's time to rock. So we tested three main predictions. The first prediction is about the decoding of the conscious content. The second is about the maintenance of the conscious content. And the third prediction is about the interareal connectivity. So I will go through the three predictions and show the final results of each of them. Regarding the decoding of the conscious content, so when we see a stimulus, like for instance the Mona Lisa, we have an experience, and we experience all the parts of this painting. We experience that there's a face, that this woman, that is slightly tilted to one side, and these are all information that should be represented in the brain in the areas that contains the conscious representation. And this is then what we were testing in the first prediction. And for GNWT, the content of the consciousness experience should involve the prefrontal cortex. So we should find all this information in prefrontal cortex, irrespective of the cognitive process. So irrespective, in our case, of whether we were showing a task relevant or a task irrelevant stimulus. For IIT, the content of the consciousness experience should be primarily located in posterior cortex. And for this prediction, we tested the category. So we were looking at the decoding the information regarding the category or the information regarding the orientation of the stimulus. So these are the two features that we tested. Regarding the category, we found that this information was represented in both posterior and prefrontal cortex. And this was true for IAG. You can see here we have on the top the posterior cortex, and we see that there was a significant cross-task representation, meaning that we were training the data on one task, for instance, the task relevant, and then testing on the decoder on the other task, on the task irrelevant. In this way, what we found, what we can see is the information that is shared between the two tasks. And we also find significant classification in intracranial EEG in the frontal areas. The same was true for MEG. You can see the posterior and frontal decoding, and for fMRI, with posterior and frontal areas both showing the codability of category. This analysis was performed by Lin, Aya, and Simon. However, when we look at the stimulus orientation, we only detect this information in posterior cortex. So here I'm showing the intracranial EEG with posterior and frontal decoding. Here is the fMRI data. As you can see, with it's in green here, you can see some significant voxels showing the information about orientation only in posterior areas, but not in frontal areas. We find a small effect in MEG, but because of the three modalities, we end up with a clearer finding, a clearer result, and we concluded from both the stimulus category and stimulus orientation decoding that the prediction from IT was passed because we were able to find both information in posterior cortex, while the prediction from GNW was only partially passed because we found the category information but not the orientation information in the frontal cortex. Okay, then we move to the second prediction, the prediction regarding the maintenance of the conscious content over time. And here the idea is quite simple. Because we're presenting the stimuli for different durations, like here is the shaded area, for instance, half a second, 1 second, or 1 second and a half, there should be something in the brain, if that area is contains the neural correlates of consciousness, signaling both the beginning and the end of the stimulus presentation, so that the experience should last as long as the stimulus is presented. That's the prediction. So for GNWT, the prediction is that there is an ignition after both the stimulus onset and stimulus offset, and this ignition happens in the prefrontal cortex, and then in between the two ignitions, there should be a silent activity state, so the neuron should stop firing during this time point. While for IIT, the prediction is that the activity in posterior cortex is sustained, so that it should be sustained as long as the stimulus is presented, as you can see from this presentation. So now we're going to test these two predictions. And this was done by Link, Alex, and I. So we found sustained activity in posterior cortex, but we didn't find an offset ignition in prefrontal cortex. So here we have prefrontal cortex, and as you can see here in the intracranial EEG, we find a nice response after the stimulus presentation. And here in the three colors, we have the three different durations, but we don't see a clear offset response, meaning a clear increase in activity after the offset of the stimulus. While in the prefrontal cortex, we find a nice sustained activity that follows the duration of the stimulus. We found some evidence in the alpha band, but we couldn't exclude that this was due of a small offset response. In this case, when we look at alpha, we should think in the opposite way. While gamma represents the activation, meaning the number of neurons that are firing, alpha is more an index of suppression. So the less alpha, the more the activation. So it's a negative correlation between alpha and gamma activity. And because we couldn't exclude that the MEG, the effect that we found in the prefrontal cortex was due to the fact that we don't have a good spatial resolution. So here, in this case, the prediction from GNWT was challenged, while the IT prediction of sustained activity passed. The third prediction is the one about inter-area connectivity. I already explained this before. One thing that we have to add here is also the temporal resolution of this synchronization between posterior or frontal areas and category-specific areas. And for GNWT, the prediction is that there is a brief and late, so after 250 milliseconds, synchronization between prefrontal cortex and high-level visual areas, while for IIT, this connectivity between high-level visual areas and V1, V2 should be sustained. So the temporal information here, the temporal prediction is linked to the prediction too. We couldn't find any evidence in favor of either theories when we look at the phase-based connectivity. So here we only found an increase in response, but after the stimulus presentation, likely due to the evoked response, but we couldn't find any increase in activity in synchronization at the time in which it was predicted by the two theories. And just to make it here, I'm showing where we were expecting to see the effects. So in terms of phase-based connectivity, we couldn't find any evidence in favor of either GNW or IIT. When we look at amplitude power-based connectivity, we confirmed the prediction of GNWT. So in both fMRI and intraocular EEG, there were prefrontal areas showing an increase in connectivity, while we didn't find this in the posterior areas. And so based on these results, so IIT is still the prediction of IIT of sustained connectivity between within the posterior thought zone is challenged, why the prediction from GNWT is partially only partially challenged. Okay, so then these are the results, and here in this table we have like we try to summarize all the results together. As you could have seen already before, we have mixed evidence in favor and against the two theories. So it's difficult to say from this evidence who's the winner, but still we know that part of the theories are correct. Like for instance, for IIT, prediction 1 and 2 confirm the fact that there is information that is persistent in the posterior cortex, sustained information and content specific in posterior cortex, while the idea that there is a maximum integration between all the areas in the posterior of the brain was challenged by the predictions regarding the connectivity. And from GNWT, there was partially pass from the prediction 1 regarding the idea that the content of the experience is represented in this frontoparietal global workspace, but not it seems that not all the aspects of the experience are represented, because we only found the category but not the orientation. While it was also partially challenged the idea of the ignition and that there is a the broadcasting of the information as a all or none threshold for the conscious experience. If you want to know more, our preprint is on bioRxiv, and you can find it by searching for the title or by scanning the QR code. And so I want to finish the presentation by talking about what's coming next. So the experiment 1 is finished, and now we are working on the data sharing. Part of the data are already accessible and can be downloaded and used to test the same predictions with different methods, or also to test other theories with the same methods, or to try to replicate our analysis and see whether there are some bugs in the code. In the meantime, we are working on the second experiment. I didn't explain the experiment before, but it's because in the first experiment we presented stimuli that are always perceived consciously, while in the second experiment we used a video game to distract the participant from some stimuli that were presented in the background in order to make them partially visible. And then we asked the participant every now and then whether they noticed the stimuli on the background, and based on their report, we classify stimuli as seen and unseen. So now we have a manipulation of awareness, and now we can test the same and also additional predictions using this different design. But also the Cogita is not the only adversarial collaboration. There are others funded by the Temple of World Charity Foundation, and here you can see 6 projects. Cogita is only one. There are other 5 projects running at the moment, testing the same theories or other theories on humans or animal models. So there is hopefully, when we will have all this information, like the results from the second experiment and from the other projects, will be way more clear what really are the neurocorrelates of consciousness. And before finishing, I want to also mention data analysis challenge that we organized the Biomech conference of this year. So the challenge is still open. So if you're interested in analyzing MEG data and you would like to test some connectivity methods that you think will work on our data set, you can just go to the website, download the data, and have a try yourself. And everything is explained on the website. But if you need, you can also reach me for more information. And that's all. So I want to thanks again all the people in Cogitate, and especially Ola Jensen, who was my supervisor during all this time, and University of Birmingham, our center, and the Templeton World Charity Foundation for funding the project. And you for your attention.